Today, I will be talking to you about networks. And in particular, I will be talking to you about networks that span huge range of length scales, like in this microvasculature, and the time scales, right? So I have length scales here, maybe a few microns, and here in this river network, it is kilometers. So what you see in these two networks is perhaps a hierarchy, right? Small vessel, bigger vessels, small vessels, small channels, bigger channels. You see some features in the architecture that perhaps are common, like loops. You see also a lot of differences between the two, let's say, styles, the two topologies. What you don't see, though, at all, is that both of these networks are highly dynamic. And you don't see this because these pictures are static. These are not movies. So in this talk, the theme of my talk, let's say, the big unifying thread of my talk is dynamics in networks, networks and chains, and how this is extremely important for the function of the network. Is something that should never be forgotten. And in fact, this changing networks, this idea that I would say networks are constantly changing was also noticed and discussed by Heraclitus or Ephesus, who said that everything flows and you cannot step in the same river twice. Now, of course, Heraclitus said it because he was alluding to the fact that the water in the river keeps changing. But I want to perhaps expand it and say that it's not only the fact that the actual matter, the flux in the river is different, but it's also the channel width. Like everything in the network is changing with time. And the form and the function of the networks, as we will see, is very intimately linked to its dynamics. Now, my group is very much interested in flow networks for a long time now, and these are some of the somewhat recent things we've been looking at that include network robustness, optimized nutrient delivery, issues that have to do with tuning of the network, optimization, topology, fluid storage, but more and more and more, we shift our efforts to understanding specifically dynamic, like spontaneous oscillations, issues of pulsatility, dynamics in development, and this is something that I will be talking to you today. Now, when I talk about flow networks, it doesn't have to be just vasculature or rivers. It could be anything that carries something, to put it simply. It could be leaf vasculature, or it could be this network in the slime mold Physarum polycephalum. This is basically a unicellular organism, macroscopic, that makes these channels, and it uses these channels to explore space and get food. It could also be power grids, for example. And what all of these networks have in common is that they can be, to some level of abstractness, they could be described with links joined at nodes, and there is a current going through the links. And at every node, I can define a net current. And what that net current tells me is whether there is fluid leaking from that node or fluid being put in that node. If the net current at a specific node is not zero, then we say that this is a boundary node. It is, I have to specify the current in or current out when I specify the boundary conditions of the problem. Then there is, of course, the pressure that is defined at the nodes. And actually, the pressure difference between the nodes will determine how much current will travel between the nodes. So I have two equations. I have the mass conservation equation, which tells me that the net sum of the currents at each node is equal to the net current S at that node. And I also have this constitutive equation, which tells me there is a function that connects the pressure drop, and the conductance, how easy it is to flow current through that link. Now, if this equation is linear, we know how to do these problems. This is Ohm's laws. This is a network, a resistor network. And we know how to do this inside and out for whatever, 150 years at least, right? So in a way, this problem is a simple problem, but it becomes very interesting once we consider dynamics. So in this talk, I will be discussing aspects of networks dynamically evolving with time. I will be discussing microvasculature and its development, and I will also be discussing the other end of the size spectrum, as I said earlier. We have the microvasculature, and we also have rivers. So I will be discussing evolution of river deltas. And in the second part of the talk, I will be actually discussing a little bit how networks actually harness dynamics for their function. I will be discussing about pulsating flows, and that will be entirely our own circulation. So this here is a relatively old movie, but I have a more recent one. And what you see initially, so this movie will loop over. So you see what you would see in a fertilized egg if you removed the top. And initially, before the heart of the embryo starts pumping, you see this not very well-defined vasculature. It's not easy to see in this resolution. And this vasculature forms a capillary plexus. And as soon as the heart starts pumping, the network starts massively remodeling. You can see it here, right? And now you have veins and arteries. And this is a much more recent movie. So you already have the big vessels, but you see how the capillary plexus remodels in this movie that loops again and again. You see it remodels and makes these higher order vessels. And this, by the way, is what you see is the chorioallantoic membrane of chicken, which is the moral equivalent of the placenta for chickens. So this is, again, stills from the movie in this older paper, and this is some drawings. And what you see here is the capillary plexus, again, disorganized vasculature when you don't have flow yet. And here, you see what happens after you have flow. Now, what happens is that the flow itself regulates the differentiation, the arterial-venous differentiation in the chick embryo. And this is due to wall shear stress. Basically, the vessels want to have a specific shear stress. And if there is high flow, that shear stress becomes higher, the vessels expand. If there is less flow, the vessels will shrink. Right? They adapt. It's known for a while that they adapt to wall shear stress. But the fact that blood vessels widen with flow has been known for several years now. It had been postulated in this thesis by Richard Thomas, who was a German physiologist. And a way to write down the equation that describes these blood vessels widen with flow is this. So we have C, which is the conductivity of a vessel E, and the rate of change of that conductivity is a positive function of the flow. But then in the absence of flow, that vessel will exponentially decay. And you notice this equation is a local information equation. Each vessel only needs to know what it's doing itself. And this equation was popularized in this paper for Physarum, which we saw earlier. But also it applies to vascular remodeling, which is this, it happens in embryonic development, but it happens in adulthood. And one could get this equation, or the moral equivalent of that equation, from the shear stress equation that we saw in the previous slide. Now, there's a problem here. And that is, in the absence of current, I would not get a capillary plexus because the steady state of this equation is no vessels, no conductance. But if I add this constant production term, then even in the absence of current, I get conductance, right? I get the capillary plexus. Now, by the way, since I wrote this full equation, if I rename the conductance something else, if the conductivity or the conductance is not conductivity and conductance, but is something that has to do with pin proteins in leaf primordium, and this current is not a fluid current, but it's an oxygen current, and so on and so forth. This equation, if I translate the fields in this equation, I'm getting the oxygen canalization equation, which is one of the accepted models for leaf vascular development. But we're not going to talk so much about this, but keep this in mind, that this equation is very, very powerful, and it appears again and again and again in development on very different systems. So what I can do with this equation is I can maybe initialize my system with some conductances. I can put in my boundary conditions. Let's say in this toy network, this is an in and this is an out. With those initial conductances, I can solve Kirchhoff. I can find the currents in each vessel. I can go here, plug in the currents, update the conductances, go back here, solve for the new currents, because if I have new conductances, I'll have new currents, and so on and so forth. Do that loop until I go to steady state. And since I want to study development during embryonic development while the embryo is growing, not just remodeling, I will make my boundary current, I will make them also increase with time, grow, right? Because as the thing is growing, everything becomes larger. You get more and more blood. And this is what happens. This is what it looks sometime after I initialize it. This is like a capillary plexus that developed, and then I have this, as soon as I quote, the heart starts pumping, as soon as you start having flow, you have remodeling, and you have a highly organized system, bifurcating system of vessels, which kind of recapitulates this transition that we saw earlier in the embryo. Now, this is a local information equation. Each vessel only knows what it's doing itself, right? And as we will see in this slide, that equation takes you to a minimum of dissipation. So here is the energy. This system has to transfer flow, right? That it is its goal. So what we care about, what the system cares about, is making it, transferring the flow, satisfying the boundary conditions with little power as possible. So here, on this axis, is the relative importance of the constant production term. It's basically how important this G term is in comparison to the adaptation term. And here is how fast the adaptation is coming. So if you're right here, to put it simply, if on this diagram you're right here, you're not really growing. This is the process that would happen in your tissue if you got an injury and you're remodeling, or if you started exercising and you needed more oxygen and you were remodeling your tissue here. This is here the moral equivalent of embryonic development. And what you see in this plot is that the final product of this developmental process, if you're making the network during growth, it has lower energy than if you're making it when you're not growing as an adult. Most importantly, I would say, is that if you're making it when you're not growing, or where you're growing with the wrong time scales, then your product, the final product, will depend on the boundary conditions. Meaning, every time you play this game, every time you grow a network, the network is going to look different. So it's not robust. Whereas here, the result is deterministic, right? You always get the same thing. You could name these veins and arteries. You could give them names. They would always come out the same, which is an interesting feature, right? If you make it when you're growing, you have a robust result. However, networks themselves, the flow, the boundary conditions, is not constant in time. It's actually highly variable. This is a movie of Physarum polycephalum, which keeps, that organism keeps popping up in this talk. And the color here basically is a proxy of how much fluid it is in the system. And you can see this, there is this, this organism does does peristaltic pumping to shuttle the fluid around. But you see it's very dynamic. And here you see a part of the mouse arterial surface network, and the color is a proxy of how much oxygen is there, and it's a proxy of how much blood goes there. So you see very heavy fluctuations. So we can take those fluctuations and put them in the model. But when we adapt with those fluctuations, we have to take the ensemble average of the currents. And these are, of course, these different currents happen because the boundary conditions change. Let's say here is the input, and the output is there, but then maybe the output wants to go there, but then maybe the output wants to go there. So I take the ensemble average of that, and I do adaptation. And what I find is that now my network is not a tree network like it was before, but it has developed loops. In fact, if the fluctuations are very strong, basically very uncorrelated and very strong, I get something that looks like the capillary plexus. And if they are very weak, I get less loops, but more hierarchy. And in fact, what I can do is that I can vary three things. I can vary the dimensionless version of this equation. And in that case, I only have two parameters. I have the time and the growth term, this G over A. And then I can vary the fluctuations. And my physiologically relevant networks would fall on an axis here. Again, it's the same equation. I just vary the coupling constant. On one side would be the tree archetype, which is hierarchical. And on the other side is the chicken wire archetype, which is the capillary plexus, right? And here, on this side, with the same equation, I get something which is reminiscent of the arterial and the venal tree. Right here, I can get the capillaries, and everything in between with a single equation. So this essentially dynamics in networks allows me to explore a very rich space of morphotypes.
Gamma is fixed by...
Yes. So the gamma essentially determines the nature of, determines how sensitive is the conductance change to the flow. We'll return to that gamma in a future slide when we talk about rivers. But I have to say this equation here, it doesn't have to be exactly this form. Somebody might complain that it's not bounded, right? The contribution of this term to this term is not bounded. I can have something like a sigmoidal. It is not, it's not so sensitive. This is a very convenient form, but it doesn't have to be just that. And I'll tell you plausible, perhaps in the discussion we can also talk more about that gamma. Okay, so now I'm going to switch gears. And in fact, what I will do is that I will step back a little bit, and I will step back by maybe 500 years or so, and look at the work of Leonardo da Vinci, who was a polymath, and he was interested very much both in rivers. This is a drawing he made of the river Arno, and in human vasculature. In fact, he has these beautiful drawings, anatomical drawings of vasculature. Now, he saw the similarity, and he discussed it in his text, and he said things like the Earth's blood is the vein of the waters. So inspired by that, let's say we're going to look at rivers. And in fact, rivers themselves, they change over time. In our lifetimes, we tend to think of them as something that it is immutable. But in fact, they change a lot. And this is maybe an example. This is the wax delta, which was actually formed due to human intervention accidentally because engineers wanted to dredge the wax lake outlet. And what happened is that right here, in a few years, we have a delta. And this is still evolving 80 years later. So now we're going to get Richard Thomas, who said that blood vessels widen with flow. And we're going to add Leonardo da Vinci, who said that rivers are like vessels. And we're going to try to understand something quite interesting. We're going to try to understand the structure of deltas right here. So we're going to take this adaptation equation, and it's kind of ad hoc, use it in a kind of ad hoc way, use it for rivers. Now, this is not unreasonable, because you understand if a channel of a river happens to have high flow, it will grow, and if that somehow you block the flow in that channel will gradually disappear. It will get, erosion will fill it in. Now, what is this gamma? So in this particular scenario, so this gamma is very important, right? At steady state, when this CDT is 0, you see that gamma predicts a scaling relationship between the flow and the conductance of the river, and the width of the river. And in fact, the scaling relationship independently, and many times, has been measured in the literature. And it's about 3/5 if we rescale it to our values here. So what essentially we're saying here is that we're going to use this effective equation that describes river evolution, and we're going to, this parameter that is actually quite important, we will make it so that the steady state of the network will obey the scaling that is observed in nature. Okay, and the question we want to answer is, why do some deltas have loops and others don't? Now, what do I mean by loops, right? So this is a tree-like delta. This is the Mekong River. And you can take a boat, and you can go here. And then you can go in this big water reservoir, sea, let's say. And you can go back up. Is that an island? We'll say no, because in our definition of an island, you have to stay in fresh water only. So this doesn't really have islands that are meaningful, whereas if you go here, you do have islands, right? So where do these loops come from? So since these deltas are tidal, we might as well go and look for the solution in the tides, right? And we're going to make a very, very simple model. I don't think you could make a simpler model than this. So here we have the source of the river, and we're going to say it's fixed. It's always the same amount. And right here we have discharge nodes. This is the ocean. And right here in this we specify a domain, and the network can evolve in the domain. But how do we model tides? We're kind of lazy, so we don't want to model the full dynamics of the tides, although we could do that. So we just say we're going to have some effective sources and sinks in the bulk of the network. And to model tidal swelling, when the water comes in, when it's like high tide, this is going to be sources of water, and vice versa. And when the tide goes down, it's going to be sinks of water, right? So this is how we impose the flooding of the river and the unflooding of the river. So we start. Maybe we start with something like this. But again, it's not so sensitive to the initial conditions. And time progresses. And we see we have this remodeling. But there is also one parameter here that determines how strong the tides are. And this is the ratio of the tidal volume of how much water is brought in and spat out by these blue nodes, divided, normalized by the input at the source. So if this number T is very small, there are no tides. If it's very large, the system is tidal dominated. It's dominated by tides, and there is no the importance of there's no water coming from the mountain. It's not important. So here I see the end results of this simulation for different T's. This is no tide. This is tide dominated. And you see, initially, for small tides, I have few loops. The color here signifies the area of the island of the loop. But as the tidal importance increases, I have more and more loops. And then for very high tides, for networks that are tidally dominated, I have no loops. And I want to compare if this is true or not. This is what I said before, but with actual data points, right? Intermediate tides, you have lots of loops. Less tides or more tides, the loops drop. So in order for us to go and test this, we go around, and by we, I mean my colleagues at Los Alamos and the University of Arkansas, and collect data from high-quality satellite images and digitize them. And these are the data set that we have. Here you see the geographic location of just 13 of these networks, but we've since I made this image, we've done much more, many more. You see the networks are all over the place. There are Arctic deltas, deltas in South America, North America, Africa, everywhere, right? So you expect a huge variation of rock, a huge variation of geography, a huge variation of conditions. And we digitize those deltas, we vectorize them, and we analyze them. We count the loops, and we know from data what is that T, what is the importance of tides. And what we see is the following. So this is the T, how important tides are. The blue is the theory, let's say, and the dots are different deltas that you see here. And you see the general trend is correct. At low tides, we have low loops. Strong tides, no loops. Intermediate tides, lots of loops. Now you're going to tell me this is very noisy. And I will tell you that's correct. So what we can do is that we can randomize the T's for its network, and we can see that if we randomize them, we get the same trend on less than 3% of the times. So this is not just statistical fluctuations, right? And the more data points that we keep putting in, the more statistically robust this trend becomes. Then you might say it might have to do with how you measure the loops. Maybe it's the loop area that matters, not the number of loops. We can measure the loops any way you want. We get the same effect. This, in fact, is the minimum fraction of channel area that must be removed to make a topological tree. This is a little bit less intuitive, but again, same effect. Now there is a consistent difference between theory and reality. And what we saw is that if we introduce noise, if we introduce actually dynamics in the on how the tides happen, a little bit more accurate dynamics, then the number of loops goes up. So it is possible, if we did a full modeling of the propagation of the tidal wave, we would have a much better fit. But the principle here is that tides is likely what is behind the architecture of the tidal deltas. And here I just wanted to show you the numerical delta, the computer delta, next to a delta with a similar T, right? So this is 0.1, and you see very few islands. Here is just you barely have an island, which is kind of elongated. This is the Wax Lake. This is Orinoco, and I put it sideways because this is also sideways. And you see many more of these elongated islands, which you also see here. Elongated islands. And here is my favorite. This is the Ras Issa, which is, I think, in Yemen. This delta is just tides. There is no water that's coming from the mountain. In this Google image, the contrast, you can see it very well. So here I have a binary version of part of the delta. And what you see is that these little leaves of the tree, they collect the water. and they take it out here and discharge it. And it is topologically very similar to this, right? So you have these little leaves that make up these trees. You see there are no islands, there are no loops that bring the water down here. So this is just one, like you can see one of these trees here. And this would be maybe this branch here. Okay, so far we have been discussing networks that dynamically evolve with time. And we have been discussing how dynamics essentially shapes their morphology, fluctuations, and so on and so forth. And now we're going to switch gears, and we're not going to talk about the permanent, irreversible change in conductance, like what was happening here. This was really network evolution, right? But we're going to discuss networks that actually harness dynamics for the function, that use the dynamics for their function. And this is going to be very specific to something which you're all very familiar with, and that is the fact that you have a pulse, right? All of you have a pulse, although this, as we will see a little bit later, this is not strictly necessary. Maybe there are some of you in the audience that do not have a pulse right now. No, you're laughing, but it's true. So this is what you see here. This is basically a human heart beating. So our flow is viscous. And in order for us to have continuous flow, we need a power source. And the heart basically is a pulsatile power source, approximately 1 watt, maybe 2, maybe, but ballpark, right? And you see it here. And that pump pushes the flow around the whole body. Now, there is a little bit of a problem because actually that heart is not the pressure differential that heart provides is not enough if you're standing up for the flow to return to the back to the heart because of gravity, essentially. So in your venous system, in the return system, in the extremities, we have a different way, an assistive system, which is composed of valves and pumping, peristaltic pumping that happens here in this cartoon from the calf muscle, right? And this is actually an ingenious system. We also have the same system in the lymph, in our lymph system, which is basically a system for the collection of waste from the intercellular spaces, and is also it functions. It assists the immune system as well. It's part of the immune system, and the lymph system, which actually, by the way, it shares a lot of in its development, it shares a lot of genes with the venous system. It also needs valves and peristaltic pumping to return the lymph. So here, again, we have dynamics. We have this dynamics being coupled to the valve, this actually peristaltic wave traveling, and the dynamics of the peristaltic waves in conjunction to the valves determines some optimal valve placement where the veins should be optimally placed. This is something I won't have too much time to talk about, but I will be focusing more in this, right? And what you see here again is the human heart pumping, but now I want you to notice the diameter of this vessel, and every time the heart pumps, this vessel becomes extended. Now, this is an effect that is very well known in the medical literature. It's called the Wind-Kessel effect, because our vessels are elastic, right? And every time the heart is pumping, the vessels increase in diameter and the pulse propagates through the system. Now, there's a problem with pulsatile flow. Now, essentially, what our capillaries need, if we take it at face value, is constant. We need a certain amount of flow to simplify it. And if we did not have a pulsatile pump, but we had a continuous flow pump, then the power that we would need to spend would be ballpark proportional to the square of that flow. But if instead of continuous flow, we have pulsatile flow with the same mean, then I need more power to push that flow. which is related to the amplitude. So the question is, why doesn't the body use the Wind-Kessel effect to eliminate the pulse? Because the Wind-Kessel effect, what it does, you can think of it as a capacitor that takes some of the flow and extends, and then let's say returns to the system. And this process basically eats up the energy of the pulse, and the pulse amplitude gets attenuated. So why haven't we built a system that destroys the pulse, replaces the pulsatile flow with continuous flow, and eventually saves energy? In fact, we kind of do it. The pulsatility decreases, right? This is a cartoon of the pressure, and as the pressure changes, in fact, the amplitude of the pulsatile wave as it goes from the aorta to large artery to small artery. So it's quite high, but as you go lower and lower and lower, the amplitude decreases, but it doesn't completely vanish. There is still signal in the capillaries, and you know it doesn't completely vanish because you take your pulse here, and you also take it here, and you feel it, and the pulse did not completely disappear, right? So why isn't pulsatility killed altogether by our body if it would be so efficient for the heart as a pump to do that? So that's the mystery. Now, biology, our own body, a healthy human body, will have a pulse. But in fact, there are people around us that don't have a pulse. And in fact, for a few months, maybe years, Dick Cheney was one of these people. So people that are in essentially they have terminal heart failure, and they're in need of transplant because it's very difficult to get a heart for transplant. There is a very large number of people that die while waiting for a transplant. And for this reason, surgeons and engineers have developed what is called the left ventricular assist device, an LVAD, which is basically a pump that is implanted in the body and takes over the function of the heart. And basically, people get this implanted, and they walk around, and they live their lives until there is an organ is found. The problem with this is that it's not a pulsatile pump; it's continuous flow, and there are engineering problems to making it pulsatile. It's much easier to make a continuous flow; it's much more robust. But as soon as this is implanted, very shortly afterwards, the patients develop a lot of diseases, a lot of vascular diseases, hemorrhaging, arteriovenous anastomosis in the gut, very, very dangerous. So you cannot have a patient for a very long time with this device because the body expects pulsatility from the pump. And in fact, right now there is research, and I think there is even some things in the market that actually have a pulsatile component to solve this problem. So now the mystery is why. So this makes us wonder, why is pulsatility so important? Is it useful for anything, at least theoretically? Not for any practical reasons. Is it useful for anything? And to do that, we have to model it. So we can start from the full Navier-Stokes which I don't show, and we have to linearize them because this is what we do, and we end up with an equation for mass conservation, which is basically an equation that tells you that a flow can be stored in an expanded vessel if needed, and there is the force balance equation that also brings in inertia. And this equation can be translated into a circuit network, to make it a little bit more clear, that's very similar to a transmission line. So this is what the equations for pulse transmission in our body would look like if we linearize them. This is the inductor, which is inertia. This is the resistor, which is, the blood is viscous. And this is the capacitor, which has to do with the fact that the vessels are elastic. So the vessel can store different amounts of volume. And in fact, I can take my original parameters here, and I can rescale things. and I can have a characteristic length scale, which depends, this is important, it depends on the capacitance or the compliance of the vessel, and a characteristic time scale and a characteristic admittance, but you can forget about this because this is not going to show up. And I can take these time scales and I can say, okay, there's a characteristic velocity. Now, is this how fast a pulse from the heart that you would show here? So there is a pulse and it's traveling. Is this how fast a pulse would travel? Let's see. So here I'm going to do the following experiment. This is a 1D vascular system. This is a spherical cow. It is connected on both ends to this power source. It's a heart. Now what I'm going to do is that I'm going to start with no flow, and at time 0, I will switch on the pump to continuous flow. And what's happened is that you have a current wave that propagates, right? And what I'm telling you here is also relevant for just sending individual pulses, right? And what's happening is that this front is going to propagate in the vessel, and there are two time scales that I need to worry about. One is this T0, which is the earliest time at a distance z that the flow will begin changing, because if I increase the flow at the pump, it will take some time for the vessel in the middle to feel that the flow has changed. Right? So I have this T0 time scale, but also you see this front that travels becomes broader and broader. And what actually matters even more than this time T0 is this time T50, which is actually the time it takes for the current to reach 50% of the final value, which is a time scale which is more robust. Now, I can make it a little bit more clear with a movie. Again, it's the same system here. This is connected to the pump, and I care about what happens in the middle of the vessel, at the distal parts. If this was a human body, here would be the capillaries, and maybe the heart wasn't doing anything, and then suddenly at time 0, it goes to a finite continuous flow. And I'm asking, how fast, after how much time, will the capillaries feel that the heart started pumping, right? Equivalently, if you have a person that their hearts had stopped pumping blood and the heart started again, after how much time will the capillaries know it? And here you see a short vessel. And what you see when this movie loops over is that you have this very fast front happening, and I hope this movie loops over. You will see this fast front, an abrupt front, but you will also see this dust line. The dust line is the mean. You see very quickly the vessel in the middle gets the mean, but you also see this front which gets reflected by the boundaries. Here, this is a much longer vessel, and you see this sharp front, which is the time T0 I showed you in the earlier slide. Doesn't really matter. What matters is like this more gradual increase of the flow in the center. Okay, so I can quantify this a little bit more, and I can calculate I can measure the normalized midpoint current. And these are for vessels of different lengths here. And it's all normalized by this lambda, which is this lambda. And remember, the softer the vessel, the longer this lambda. And you see for this short vessel, the blue one, I have all these reflections. And for this long vessel, I asymptote like this. On this axis, it's time, but it's time normalized by this beta. What is this beta? Well, if I were to measure the residual current, because what I care is the final current, so I can go here and measure the difference of the current to the residual current, right? And I plot it in a log plot. I see everything falls quite neatly on a straight line. So I can extract from this straight line beta, which is like it's something that we call the response scaling function. It tells me how fast will the vessel adapt to the new boundary condition. This is not a permanent irreversible adaptation of the diameter, but it is just the vessel responding and assuming the flow that is imposed by the boundary conditions, like the steady, how fast the vessel goes to the steady state flow. Here is this beta, which actually for the single vessel, this case, it can all be done analytically, which is great. So we have right here long vessels, or if you keep the vessel length constant, it's equivalent to have vessels that are very squeezy, very compliant because of this normalization of this axis with this lambda. Okay, so here essentially you can say that you have either long vessels or compliant vessels. Here you have stiff vessels or short vessels. And what this plot tells us in simple is that soft vessels take much longer to respond, to adapt, to adapt to the new conditions. Stiff vessels take much shorter time. And so we can do it analytically for one vessel, but for complicated networks, and these are just cartoons, we can do many more generations than this. It's just not really nice to plot. It's the same story, right? Large networks, and this is what here I mean by large. It's this path integral. Large networks take longer to adapt than short networks. Or equivalently, if you keep the network the same, squeezy networks take longer to adapt. And all of these data points that you see, all this craziness, is just different architectures. You can have different architectures over here. You can have loops, no loops. You could have different scalings or how the networks bifurcate. You can have noise, no noise. It's surprisingly robust. This slope is surprisingly robust. What's not robust exactly where this is going to happen. So the curves here, the network size, it looks like there is a characteristic network size that depends on the architecture, on when you shift from flat to sloped. And now I can go and measure something else as well, which I care about, and that is dissipation. So if you remember initially in the talk, I said wouldn't be nice if the wind kessel killed pulsatility. Because if the wind kessel killed pulsatility, then I would need less power to circulate my flow. And I also said that if you have very compliant vessels, you can kill pulsatility early on. Great. So this is where we are. These are compliant vessels here, the red. And that is low dissipation. Right? And this is dissipation versus this response scaling time, how fast you adapt. So here, you have low dissipation, but it takes you a very long time to respond. Right here, up here, it takes you very little time to respond. You're very stiff. It takes you very little time to respond, but it's very high dissipation. And again, this trend, this trade-off is very robust for many different networks. Yeah. So you can ask me, where are we as humans on this plot? Are we over here or are we over here? So this is something that one has to be very careful, right? One has to be very, very careful once you talk about humans and real living beings. So we of course, we wanted to ask that question because this was all theoretical. So where are we on that plot? So for that, we went to this wonderful paper, which is basically a model of the entire adult circulation. This was a lot of work by Minard and Smolitz to acquire, where basically they have an entire map of all human circulation. And they use a very complicated nonlinear model to see what happens to this to the system when you have, when the heart is pumping, and this is the cardiac output, right? So what we did is that we took their model, but used our linear equations with the same input and output over here, and then compared against their predictions and against actual measurements when they can happen. And the linear model is not that bad. So right here on this side of this plot, so here, for example, is mean flow, and each dot is a vessel, and they are basically presented on this line, although our system is not linear, right? So right here is the Venus system, and we don't get it very well because we don't have valves in this model. And right here, I think this is the head, and we fail completely, especially in the face. But OK, we don't care about the head for now. But you see, it works relatively well, this linear model. And when we do, and this is very preliminary, but yeah, maybe I should say it, but not show it as a slide. It looks like we're somewhere around here, in this interesting point, and we have no idea why. Yeah. Anyhow, so let's go back to this importance of time, right? So what the time that I've been talking about, this response function, tells us is that in the absence of any neuronal, which is active input, the network can adapt to changes in the boundary conditions. Let's say suddenly my heart rate increases or I go for a run, right, with finite speed. Which is roughly, for our human body, as we calculate, 4 to 5 seconds, basically. That's not, however, that is a lower limit, because in fact, we're integrated with with an amazing neuronal system, which basically can transmit information faster than this mechanical limit, in a way. But in the absence of neurons, the beta, the response function I was talking about earlier, would be the limit on how fast our body would adapt, which is actually also how mechanical information is integrated in our body. If I squeeze here, how fast will the rest of my system know that I squeezed, right? Or if I start losing blood here, how fast will the rest of my system know that this has happened? Now, what role could this mechanical, this speed of mechanical information transmission could have played in evolution? This is, again, another very interesting question. So I'm reaching very quickly the end of my talk. So this is a summary, I would say. So I talked to you about both irreversible changes in the network due to dynamics. This is network evolution. And in the second part, I talked to you about reversible changes, right? Dynamics in two different ways: dynamics shaping development and dynamics determining function. And you saw how pervasive this dynamics is. Now, this is a little bit of philosophical thinking that I personally find very interesting. And that is that it seems that these biological flow systems, and let's forget about rivers for a while, the development relies on this mechanical information to happen. And in fact, from what we know about LVADs and about what happens to people that don't have a pulse, it looks like our body expects pulsatility to maintain the vessels. And once you take this pulsatility away, once you take this expected dynamics away, the network starts remodeling in very pathological ways. And we don't understand at all yet why. We just observe it very sadly in patients. It looks like the steady state has evolved to anticipate dynamics and to respond to it in an optimal way. And of course, all of these ideas perhaps would matter for other networks that are maybe mass-preserving, like ecological networks, whom, right? Which are not, again, they're not, a lot of times we study them as, as networks that don't have a serious time component, but well, not always, a lot of time, right? But in truth, they are very, dynamics plays a huge role, and also, similarly, metabolic networks. So with that, I would like to thank my group. Unfortunately, we don't have a recent picture. This is from May 2020, and everyone looks sad. This is even earlier than that, and there's been a lot of changes, but I thought I would show it because a lot of the work that you see was done by Henrik, Ron Ellenfitz, Sean Fancher, and this amazing undergraduate, Adam Konko. Okay, so yeah, thank you very much for your attention, and if you want to contact me after the talk, this is my email to continue the discussion.